All right.
Okay, great. So yeah, I think we have a good number of people joining here. So it is my great pleasure to introduce our speakers today. So today we welcome Dr. Luis Barreiro. He's an associate professor at the University of Chicago, and his research primarily focuses on understanding how natural selection has contributed to the evolution of our species, and particularly looking at the evolution of immune responses, both at the interspecies as well as between different human populations. And so he's published a lot of very fascinating papers, particularly some recent work on the association between ancestry and differences, genetic differences and immune response. And we're all really excited to hear more about his research today. Thank you.
All right, thanks for the introduction. So like Linda just mentioned, like in the lab, we are very interested in understanding the relationship between pathogens and the evolution of the human immune response. And I'd like to just try to make the point that throughout human evolution, pathogens have been really probably the strongest selective pressure that humans have to face. And I think these days it's easier to convince people of that after these 3 years of pandemic that we just endure. But probably the best way to actually appreciate the impact of pathogens during human evolution is simply by looking at survival curves, historical survival curves. And as you can tell here from this plot, so in this plot in the x-axis, I'm plotting the age and survival rate in the y-axis. And for most of our evolutionary history, whether you compare the people living the Neolithic up to the 19th century in the UK, these curves have hardly changed. And by age of 20, 25 years old, 50% of the individuals will die. Then in a very short period of time, we became much better at managing the detrimental impact of pathogens through the advent of things like vaccines and antibiotics. And suddenly we start seeing survival curves that look like this. So I think this is a very good illustration of the detrimental impact that pathogens had to human lifespan and therefore selection. So the way, traditionally we have been trying to identify these signatures of natural selection imposed by pathogens is by using classical population genetic tools that look at patterns of genetic diversity. And that's actually how I started addressing these type of questions. And the reason why we can do that is that when you have natural selection acting on a particular variant that, for example, increases fitness and protection against a particular pathogen, that selective event is going to leave a very particular signature in the genome. So what will happen, imagine that this red mutation protects you against a certain pathogen that is endemic in your area. So individuals that have this variant and the haplotype that lies with this variant are going to be have increased fitness, and therefore this variant increases very rapidly in the population. And what will happen is that the patterns of genetic diversity before and after selection are going to be very different, and you can identify these signatures of selection by means of what we call neutrality tests. So when you apply these neutrality tests genome-wide, what we see is that both at the interspecies level as well as within human populations, genes evolved in immune processes tend to be highly enriched for these signatures of natural selection, which really provides further support to this notion that pathogens have been a really important selective pressure. However, one limitation of this type of approach is that they are only looking and relying on patterns of genetic diversity. So there's no actual connection between a signature of natural selection as identified by these neutrality tests and the natural phenotypic outcome. So therefore in the lab, the way we kind of start thinking about addressing these questions is by start thinking about population diversity in immune processes, not only as a function of this genotypic diversity, but as well as a function of phenotypic diversity in the population. So the way we kind of try to integrate all these different things is that in addition to obtaining genetic information from individuals from the general population, we also collect cells, and in this case, it's always going to be healthy individuals. So we really care about understanding variation in a healthy population, in the general population, and then when we take these cells, we infect them with whatever pathogen we are interested in, characterize the way different individuals respond to these pathogens, And if we are lucky enough, we are going to be able to identify genetic variants that explain this variation in immune responses that I'm going to call immunological quantitative trait loci or immunological QTLs. And then once we have these variants that already we know a priori to be functional, we overlap those with signals of natural selection. And when we see those overlaps, at that point, not only we can understand, not only we can claim that there has been selection that acted in a particular gene or genetic variant, but also understand what's the function that has been selected for. So kind of using this general approach, one of the things that we've done in the past was try to understand what was the contribution of potential contribution of natural selection to population variation in immune responses using this framework. And what we did was to collect cells. In this case, we focus on a particular cell type called macrophages that are very important innate immune response, very important cells for the innate immune response to pathogens. And we collected those data on a panel of 175 individuals with varying degrees of European and African ancestry. Then from these macrophages, we infect them with 2 live bacteria, Listeria and Salmonella. And the readout of this experiment at the time was just bulk RNA sequencing on non-infected and infected cells. So what we saw at the time was that over 20% of the genes that respond to either infection, they show a significant difference in the intensity of the response between European and African ancestry individuals. And interestingly, what we saw was this very striking trend whereby individuals with increased African ancestry tended to engage a much stronger transcriptional response to infection, particularly when focusing on genes involved in inflammation. So the other thing that we did is that by combining genetic information from the same individuals, we were able to estimate that about 30% of the population differences in immune regulation are explained by changes in the frequency of these expression quantitative trait loci, or in other words, genetic variants that have an impact on variation in gene expression levels. So now we basically, going back to this general framework, we have now a set of genetic variants that we know to explain differences in immune response across different ancestry groups. You can ask whether they are enriched for signals of natural selection, and that's indeed what we have actually seen. So this figure here just shows in the top the results for signals of natural selection in Europeans, and in the bottom in African ancestry individuals. So the y-axis is just a statistic that measures indicative of recent positive selection called IHS. And basically, the only point that I'm trying to make is that all these genes that are highlighted here above this dashed line are genes for which we have a genetic variant that has an impact on gene expression levels, and it is highly differentiated between European and African ancestry individuals, and also shows a signature of recent positive selection, providing evidence that basically natural selection has contributed to present-day difference in immune response between different ancestry groups. So this was a kind of the first work that we did back in the days, and more recently we got interested in trying to see whether these patterns hold true across other immune cell types, as well as with respect to other pathogens. And specifically, we got interested in thinking about viruses, because recent data suggests that viruses across the different types of pathogens that exist, that viruses have probably been one of the strongest selective pressures throughout human evolution. So again, this paper was published later in 2021, led by PhD student in my lab, Ailey Randolph. So I'm just going to really summarize this in 2 slides, and because today I decided to primarily focus on unpublished data, so it is just kind of a vignette of what we found, but I invite you to then go and read the paper in much more details, because there's a lot of things that I'm not going to be talking about. But basically, in a kind of a nutshell, like the experiment was quite similar to what I described earlier for the macrophage experiments, but this time around, we started with a panel of 90 individuals, again, with varying degrees of European versus African ancestry. And this time around, instead of focusing on an isolated cell type, we took all the PBMCs, so peripheral blood mononuclear cells. So this composes, all the key immune cell types, B cells, T cells, and etc. And then infected those PBMCs with the influenza A virus and performed single-cell RNA sequencing on infected and non-infected cells. So this is kind of the mandatory UMAP when you do single-cell RNA sequencing, just showing that We identified all the main cell types that we expect to have in PBMCs, natural killer cells, CD8 T cells, CD4 T cells, monocytes, and B cells. And the entire data set was about 230,000 cells across these 90 individuals. So what did we find? So confirming what we had seen in the macrophage experiment and in response to bacterial infections, we also see that in response to a virus infection and across all these different cell types, there are hundreds to thousands of genes that for which we see a linear relationship between genetic ancestry levels and gene expression in both infected and non-infected cells. And one of the interesting things that we saw that we could learn from this study, which had been impossible in the past when we were just doing bulk analysis in a single cell type, is that a lot of these ancestry effects tend to be cell type specific. So we estimate basically 52% of these what we call population differently expressed genes, or POPDE for short, were only identified in one or two of these immune cell types, really highlighting and stressing the importance of characterizing these effects across a large array of different immune cell types and likely different conditions. So in terms of what did we learn in terms of the biological patterns that are the most divergent between European and African ancestry individuals, we saw this very striking pattern where European ancestry individuals tend to engage a much stronger interferon response to influenza A infection. That's what I'm trying to show here in this plot. Is the x-axis is just a quantitative measure of the levels of African ancestry for each individual. So we never, in all the analysis that I'm going to be talking about today, we never binarize individuals as being African versus Europeans. We actually use genetic information to estimate what's the proportion of their genome that is of European versus African, and we use those quantitative metrics of genetic ancestry to correlate with whatever metric we are interested in. So in this case, I'm correlating those levels of genetic ancestry, in this case, African genetic ancestry, versus a score that I call interference score, that you can think about it as how active is your interferon pathway in each of these individuals. And specifically when focusing on infected cells, and those are like the filled lines, so not the dashed lines, you see this striking relationship where individuals that have increased European ancestry, so basically reduced African ancestry, tend to have a much stronger interference score as compared to individuals that have increased African ancestry. And then I'm not showing you the data here, so you'll just have to believe me. But then we also show that this stronger initial interferon response is predictive of a better ability to limit viral replication at later time points. So if you actually measure viral titers in the cultures 24 and 48 hours after infection, if you engage a stronger interferon response early on, you also control the virus much better. So the other thing that I'm not going to show you, but it kind of relates with what we have seen before in the context of immune responses to bacterial infections, that again, if you combine these transcriptional differences, these ancestry-associated differences in immune regulation with genetic information, we again estimate that about 30% of the differences that you observe across these different cell types appear to be linked to these EQTLs that vary in allele frequency between European versus African ancestry individuals. So in other words, if we just combine all these different data, we can conclude several things. First, there are clearly significant differences in immune regulation between different ancestry groups. Second, genetics does contribute significantly to those differences. And third, natural selection seems to also have played a role. But if we think about the contribution of genetics, though it's clearly important, it still only explains about 30% of the differences. So in other words, there are hundreds to thousands of genes for which we see a very significant relationship between genetic ancestry and, say, gene expression, or the magnitude of the response to these infectious agents, and yet we cannot link those differences to any particular genetic variant. And of course, for the vast majority, what this is going to tell us is that those differences are not genetically driven and just reflective of the fact that genetic ancestry itself is going to correlate with a variety of other environmental factors and social determinants, and we are probably likely capturing those effects.
Luis,
Go ahead.
Did you look at locus-specific ancestry for any of these genes as opposed to individual EQTL variants?
So we have not done. So all the stuff all the results that I showed was using like genome-wide ancestry estimates, not local ancestry estimates. But that's something that of course makes a lot of sense. And my guess is that if we do local ancestry estimates instead, that we will probably actually have larger number of ancestry effects, given the fact that we are seeing this, clear importance of cis-regulatory variation. So if you focus really on the ancestry around the locus, my guess is that we will see even stronger effects, but we haven't done that yet. So one of the things that we got interested, so from this point on, everything that I'm going to show is like it's unpublished data. So one of the things that we got interested is to try to understand what might be the contribution of epigenetic variation to these ancestry effects, and that's motivated by a number of things, including the fact that we know that at least in vitro, epigenetics can have actually a very important role at determining the magnitude and nature of the innate immune response that we engage. So, for example, we know that if you cells exposed to particular immune triggers, such as beta-glucan or even BCG vaccination, they become primed in such a way that if you restimulate these cells, these innate immune cells, with a second challenge, they are going to respond much stronger to a secondary immune challenge. So in other words, your exposures, even in the context of an innate immune response that supposedly has no memory, but we now know that even in that context, whatever you're exposed to, it's probably going to shape the epigenome of your cells in such a way that might impact your secondary responses. The second thing, we know that also in vivo, this appears to be true. So we have done in the lab showing that if you vaccinate mice with BCG, what will happen is that so BCG is the vaccine used against tuberculosis. So the BCG has this ability to actually reach the bone marrow and alter the transcriptional and epigenetic profile of the stem cells in the bone marrow in such a way that then you're going to generate in the periphery and in the tissue macrophages that are much better able to control a mycobacterium tuberculosis infection. So again, suggesting like how exposures can have an impact on your epigenome that ultimately impact the function of your immune system. And then finally, in the context of epigenetic variation, we also know that genetics also plays a crucial role at determining the epigenetic variation across individuals. So the hypothesis becomes like, can epigenetic variation, whether driven by environmental factors or genetic factors, also contribute to inter-individual variation and ancestry-associated variation in immune responses? So to address that question, we did the following experiment. So this is work led by a PhD student in my lab, Katie Aracina. So we took, cells again from 35 individuals. So here we are again focusing on monocyte-derived macrophages, so an isolated cell population. And in these cells, we performed basically, an extensive array of molecular characterizations ranging from whole genome sequencing, RNA sequencing for gene expression, chromatin accessibility, the characterization of 4 histone marks, H3K27 acetylation, ME1, K4ME3, and K27ME3, as well as whole genome bisulfite sequencing. So it's a large data set with over 200 billion reads, and we actually made all this data available in this browser that we built, where you can just look for any gene, any SNP of interest, and you'll get a lot of information about the epigenetic profile of these cells before and after. Sorry, I think I forgot to mention something important. So we looked at these profiles before and after infecting the cells with flu, with the influenza. So again, we are really looking at trying to determine how this epigenetic variation impacts your response to flu. So there's a lot of data and a lot of different data sets. So I have no time to actually go into the quality control of the data. So the level of confidence that I'm going to give you that the quality is very good is going to be this. You just kind of have to believe me that it's, that we did all what we could do and the data is really, very, very good. So the first thing we wanted to know, of course, is just like, how are these different epigenetic marks responding to this infection? And what we saw is that, gene expression, chromatin accessibility, and H3K27 acetylation, which is a marker of activation, if you will, are by far the most dynamic regulatory elements in response to flu. So, close to, if 50% of the peaks/genes are altered, for these marks are altered in response to infection. By far, also, the least dynamic responses to flu were observed for all the repressive marks, and this includes H3K27Me3 and DNA methylation, for which, only a very tiny fraction of the genome responds to flu infection. So when you look at what pathways are being up or down regulated in response to flu, we see what we would expect at the gene expression level. We see very strong upregulation. So anything that is dark, orangey or whatever color it is, it's going to be upregulated, and blue it's downregulated. So you clearly see what you would expect in response to a viral infection. You see very strong upregulation at the expression level of the interferon pathways, inflammatory response, etc. And what was interesting to see is that when you look at the genes nearby the chromatin accessibility peaks that change, the acetylation that change, etc., you see basically a pattern that mirrors what we see at the gene expression level. So you also see increased in accessibility, increase in acetylation, increase in H3K4Me3 marks in the same exact pathways that you see for gene expression. And kind of a nicer and probably easier visual for what's happening. I'm just showing you like the patterns for a single locus, so the region encoding for NF-κB, and you see at the gene expression levels, see very strong upregulation of the gene, and this is accompanied by the gain or the opening of the chromatin and the gain of acetylation marks in putative enhancer regions as marked by H3K4Me1 that also increases. And then we overall see a decrease, basically decreased levels of H3K27Me3 levels that is a repressive mark. So all these changes are clearly highly coordinated between the epigenome and the transcriptional response. So, but like I said, one of the things that they are really interested in is understanding, to which extent this overall and shared mechanistic response to infection varies as a function of genetic ancestry. And so to do that, as before, we collected genetic information. In this case, we did whole genome sequencing. So there's all these individuals have been sequenced at 30x coverage. And then we use that information to calculate the levels of European versus African ancestry for each individual. And now what we're going to be doing is what I have here in the bottom. So we're going to try to identify genes that I call population differently expressed genes, or PICs. So PICs refers to the epigenetic marks, of course. There's also CPG sites, but I didn't want to put that many slashes here. But the idea is exactly the same. So we are basically looking to try to identify genes or peaks for which we see a correlation between, for example, gene expression levels or peak intensity and the quantitative levels of African ancestry, for example. So this is the POP-DE, and then we can do the same thing, but now replacing the y-axis by the log-to-fold change response. So instead of being, for example, gene expression of NF-κB, it's going to be how much did you upregulate NF-κB in response to flu. That's going to be your phenotype, right? And these are what I call population differently responsive genes, or POP-DE. So I'm focusing on POP-DE effects. These are kind of the numbers. So the absolute numbers are a bit hard to compare, because of course we are analyzing about 13,000 genes for gene expression, but for example for H3K4Me1 we are about 100,000 peaks across the genome. So it's very hard to compare absolute numbers. So the y-axis in this case is basically the proportion of features that are significantly associated with genetic ancestry across the different marks. And probably, as you can tell, there's like thousands of peaks and genes that correlate with genetic ancestry. But probably one of the most interesting patterns that we see is this one whereby ancestry-associated differences in gene expression appear to be primarily associated with variation in enhancer activity as opposed to variation at level of co-promoters. And that's kind of illustrated by this contrast between, 20% of enhancer-associated H3K4ME1 peaks are classified as POP3E versus only 1% of the H3K4ME3 peaks that primarily mark enhancer promoter elements. So we see this kind of larger variation and larger correlation with genetic ancestry at the level of enhancers as compared to core promoter regions. So when you look at population differently responsive genes, we see that there's about 21% of the genes that respond to flu show a significant difference in the intensity of the response. And here in the bottom, I'm kind of trying to summarize those results into particular biological pathways that are, of course, critical in the context of an immune response. And the way I'm showing you here is basically for each of the pathways. So focus, for example, in the interferon alpha response pathway, each dot corresponds to a score that tells you like how much this individual upregulated interferon. Well, sorry, the levels of interferon alpha. Let me step back. Actually, it's the response. So like how much this individual upregulated interferon alpha in response to flu. And then I'm just kind of making the contrast between individuals of European and African ancestry. So the p-value is actually reflecting the correlation between ancestry and the response, so the linear relationship through a linear model. For visual purposes, I'm showing it as a box plot by binarizing African versus European, but just that none of the analyses were actually then binarized in terms of the statistics. But you clearly see that, across many of these pathways, you see significantly differences in the response between European and African ancestry individuals. And those are, again, centralized, consistent with what we have seen before in past work in things like interferon response, as well as inflammatory responses, TNF alpha, VNF kappa B, etc. So you see quite a lot of differences in these pathways in the magnitude of the response as a function of genetic ancestry. So this data now also provides a kind of a nice opportunity to start asking the question: can your baseline epigenetic levels predict your response to infection at the gene expression level? So that's what we try to do using this, use some machine learning. So we use elastic net regression to really assess the predictive power of these baseline epigenetic levels to the transcriptional response of this pathway. So now I'm trying to predict these values here, these numbers that I showed in the plot before, just based on baseline epigenetic information. And what this kind of colorful M&M plot shows is you can think about each circle and each number here corresponds to the correlation between the predictive and the observed value of each mark for each pathway. So for example, the 0.8 that I'm highlighting here with my mouse says that using baseline levels of H3K27 acetylation, I'm able to predict with 0.8, correlation of 0.8, the interferon alpha response, right? And what you can take from these figures is that across all these different pathways, there's at least one epigenetic mark that is strongly predictive of the response. And these kind of just some particular examples. So for example, here clearly what I'm showing you for interferon gamma, here is just the predicted score versus the observed score. And you see that we are doing pretty well at predicting these responses, these transcriptional responses, just based on baseline epigenetic levels, supporting the idea that, an individual's response to infection, or at least flu, is driven at least in part by the epigenetic landscape of the genome prior to the infection. So now we wanted to understand, to which extent, again, these differences are genetically controlled. So we went back to doing, our favorite thing, that is QTL mapping. And again, just for those that are not familiar with what QTLs are, just kind of a reminder again. So what we are talking about, imagine this scenario here that you have a gene, toll-like receptor 4, that there's a mutation, say in the promoter region of this gene, that you can either have the G allele or the A allele. And now assume that the A allele disrupts the binding of a transcription factor that is important for the activation of this gene. And what will happen now is that if you just look at the variation, at the relationship between genotype for this SNP and gene expression levels in the population, what you would expect to see is something like shown here in this plot, whereby individuals that are homozygous for the A allele that disrupts the binding are going to show on average lower expression levels of toll-like receptor 4 as compared to individuals that are heterozygous or homozygous for the G allele, right? So we can do this now basically genome-wide across all the genes and looking at relationship between the expression levels of those genes and all the cis-genetic variation around them. And in this particular case, we are going to be doing that not only for gene expression, but for all these epigenetic marks that we measured, that we collected data on. So when you do that, again, we find thousands of genes and features, and by features, I'm really kind of broadly speaking about all these different epigenetic marks for which we find this relationship between genetics and variation in those marks. And you know, I'm skipping a lot of details in this plot. As you can tell, there's like things like STR, specific STR stands for short tandem repeats. So it is just like in this, because we had whole genome sequencing data, we actually look at the relative contribution of tandem repeats versus SNPs to these QTLs. And one of the things that we found is that actually there's a lot of QTLs that are only found if you look at these short tandem repeats that we would have missed if you just look at SNPs. That's not the point of today's talk, but that's what this figure is trying to illustrate. The point being that there's a lot of genetic variation associated with variation in these molecular traits. And the other thing that we also see is that similar to what we see at the gene expression level, many of these epigenetic QTLs are also only observed in a condition-specific manner, and notably, for example, when you infect cells with flu. So there's a lot of GBA interactions also at epigenetic level, where I'm showing you two examples here. For example, this here that I'm highlighting in purple, and like with my mouse, it's on top of it. You see it's a flu-specific, flu-infected specific chromatin accessibility QTL. You see that this SNP is not really significantly impacting chromatin accessibility levels prior to infection, but after infection, you see an association. And then in yellow, it's an example of a flu-specific methylation QTL. You see no association prior to infection, and once you infect the cells with influenza, you see this clearly strong association between genotype and methylation levels. So now that you have like this kind of catalog of variants that are associated with variation in these molecular traits, we can start asking the question, to which extent are these genetically controlled differences in epigenetics explaining the ancestry effect that you observe? Well, actually, I'm actually skipping something, so just let me step back for a second. So something that we also noticed by looking at these different EQTLs along this regulatory cascade is that a large fraction of these QTLs are shared across epigenetic marks. So in other words, what I'm showing in this plot is, for example, in the first column, if you just take all the QTLs, so those illustrated by RNA-seq means like if you just take all the QTLs and just ask whether those same SNPs, those QTLs, also tend to be, for example, chromatin accessibility QTL or H3K27C QTL and whatnot, what we see is that 50% of those QTLs, they overlap with at least one other epigenetic mark. And that's true across all the epigenetic marks. And if you just, you know, another way to think about it is like this. So for example, if you condition on EQTLs and then just ask how many of those EQTLs are also, for example, chromatin accessibility QTLs or H3K27 acetylation QTLs and whatnot, you see that, for example, among EQTLs, about 40% also impact chromatin accessibility, about 25% are going to impact H3K4 ME1 levels, etc. So there's a lot of sharing of these different regulatory marks across across all these across basically the entire epigenetic landscape. So going back to what I was trying to say about trying to connect these EQTLs with population variation in these epigenetic marks, what we can do is just do something relatively simple. That is like just ask the question about what's the relationship between the predicted differences that I will how. So if you just have the QTL data, you know the effect size of the QTL, and you also know what's the differences in allele frequency of these EQTLs between populations. So just based on genotype of our individuals and the EQTL effect size, you can predict how different should be how large should be the difference between European versus African ancestry individuals if those differences were only purely genetically controlled. And then you can correlate those predicted values based on genetics alone versus the observed values. And if the correlation is strong, that will tell you like how much, what's the fraction of the variance that is predicted by genetics. So when you do that, for example, for a TAC-seq, you see that the R square is 0.52. So in other words, what this is telling us is that the basically genetics explains 50% of the variance in chromatin accessibility between European versus African ancestry groups. And you can do this across all the different marks. And what we see, just kind of a summary here, is this overall pattern where actually across the board, genetics plays a more significant role at explaining population difference in epigenetic marks than it does actually at explaining differences in gene expression. And I think that's just reflecting the fact that probably gene expression is more downstream of all these epigenetic marks, and it's of course affected by probably the combination of all these different regulatory elements, where things like the TAC-seq chromatin accessibility is probably more directly impacted by things such as differences in transcription factor binding sites. So what about trying to understand what pathways, to which extent these genetically controlled differences explain the differences in these pathways that I mentioned earlier. So these are just kind of the enrichments that we saw. So the blue means that these pathways showed increased activity in individuals with increased African ancestry, and red means the opposite, that the pathway showed increased activity in individuals with increased levels of European ancestry. And this is kind of the matrix that you see in the original data, right? And now what we can do is just kind of regress out those genetic effects and see how does this look after regressing out these genetic effects. And as you can tell, a lot of these enrichments just go away. So in other words, it suggests that a lot of the enrichment that we are seeing and the differences in immune regulation are at least in part regulated, controlled by genetics. And if we focus on particular pathways that consistently come up as being significantly different between European versus African ancestry individuals, such as inflammatory response pathway, you see that there was a lot of enrichments prior and after, almost everything goes away. And if you now do the same thing, just looking at the response itself, in the original data, I showed you before that there was a significant difference in the response between European versus African ancestry individuals. When you do the same thing now, regressing out all these genetic effects, you see that that difference goes away, suggesting that a lot of the differences in inflammation that we see between ancestry groups appear to be genetically driven. So in contrast, when you do the same exact thing for interferon responses, you actually see no effect whatsoever. So basically the data remains almost the same, suggesting that though we see strong relationship between interferon response and genetic ancestry, and we saw that in our single cell data before, we see this again in this data, those differences do not appear to be the result of cis regulatory events. So they could still be genetically driven, but that will have to be through a different mechanism, such as a trans effect or multiple trans effects that we are not being able to capture. But most likely, what this might reflect is that these differences are, probably the reflection of environmental factors or other factors that correlate with genetic ancestry that we are not able to measure. The other thing we wanted to understand is are these QTLs, these epigenetic QTLs, potentially informative at trying to understand genetic susceptibility to disease? and particularly focusing on immune-related disorders. So we did, the obvious thing, that is to do colocalization analysis between these different EQTLs and the GWAS variants for these different immune traits listed here. And when you do that for RNA sequencing, so just for EQTLs, we do colocalize a number of GWAS hits, but the numbers are relatively small. And what's interesting that when you do the same thing now for all these epigenetic EQTLs, you see that the numbers explode quite a lot. And actually, across the board, like if you just take the union of everything that colocalizes with GWAS hits, only 7% actually are EQTLs. The vast majority, so basically, the remainder, 93%, are genetic variants that for which we find an association with epigenetic variation, but not with gene expression levels. And this is shown here, an example for Crohn's disease, where you see this variant that is a GWAS hits for Crohn's disease that is colocalized with the chromatin accessibility QTL, but does not colocalize with any QTL in the region. So what does this mean? Like, one way that you can think about it is that, many people do this, like, you do like a QTL for any new molecular trait or whatever, and then you kind of find a lot of things that colocalize with that trait, and you're going to say, okay, this is potentially a new mechanism or whatever of disease susceptibility. I don't think that's necessarily what's going on here, because it's, at least maybe I'm not creative enough, but I have a hard time imagining like how, for example, a chromatin accessibility QTL without ever being an expression QTL could be a mechanism, right? So I feel that at some point we have to pass through changes in gene expression to actually ultimately have a mechanistic path to disease susceptibility. So I think what these epigenetic QTLs are doing is that they are representing very good proxies of EQTLs that do exist, but they exist in an environment that we have not yet captured, right? So in other words, imagine that you are in the right environment, you have your EQTL location here in the triangle, and say that this site disrupts the binding site of a transcription factor. So what might happen is that this pioneer transcription factor, for example, is going to bind more or less depending on the genotype, and that will directly impact levels of chromatin accessibility. So we'll have a chromatin accessibility QTL. But then these enhancers say that it's only active in a particular environment because you need the recruitment of an additional transcription factor here in yellow. And so if you capture it in the wrong environment, you might not see the QTL, but you still will see the epigenetic QTL. So in other words, I think just kind of an interesting and a potential good way to capture these condition-specific QTLs, and it might help us get closer to the potential mechanism. Okay, so
Luis,
just to kind of go ahead, please.
Yeah, sorry. Is it possible also that timing is a really important factor here? That basically all of our EQTLs are coming from kind of one time frame, one time window, and that many of these things are sort of acting in a very dynamic way, and we're just not good at capturing dynamics.
Absolutely, yeah. So I think I'm kind of using the term in a specific environment very loosely. So basically, that could include like I said, time effects. It could include different infection effects. It could include like even potentially cell types. So that's actually part of the challenge, that there's there's an infinite amount of environments that one can look at. So you will never be able to cover all those environments. So I think the point I'm trying to make is that maybe a way to circumvent that limitation is that these epigenetic EQTLs might actually be good proxies of those environments that we will never be able to capture, right? So.
Sure.
So kind of to wrap up this section, because I'm afraid that I'm going to run out of time, and I would like to just tell you a little bit about the other story, is that so we do see that genetic ancestry effects on the immune response to flu are common and affect several immune pathways. Baseline epigenetic profiles are strongly predictive of the way you're going to respond. By combining, all these different differences in molecular traits with QTLs, we show that the epigenetic ancestry-associated difference in the epigenetic landscape are largely genetically controlled, actually more so than variation in gene expression. And finally, that these epigenetic QTLs might help, contribute to fill the gap between these GWAS kits and understanding potential disease mechanisms. So the other thing that I like to go back to is my introduction. That is, our motivation is really try to understand also selection and the impact of pathogens to the evolution of the immune system. So we have shown before that these EQTLs that are associated with variation, ancestry-associated difference in immune response, tended to be enriched for signals of natural selection. So now we did the same thing for epigenetic EQTLs, and this is a kind of a summary of those results, where in the x-axis I'm just showing you evidence for recent positive selection as measured by IHS, and the y-axis just shows the levels of population differentiation as measured by FST. So a large value will be indicative of also local positive selection potentially. And what we see is that when you look at the combination of these two statistics, 70% of all the colocalized SNPs, all these colocalized epigenetic QTL show a signature of natural selection. And for the vast majority, with the exception of 2 of these hits that are here, they are all only epigenetic QTLs. They don't seem to impact gene expression levels. Again, suggesting that these epigenetic QTLs also have an impact on organismal fitness. So, but anyways, the point I'm trying to make is that we are seeing all these signals of natural selection that we predict to be imposed by pathogens. But one of the challenges is to actually link these signals of selection with a particular pathogenic agent. And the reason why is that these immune pathways, of course, are function in response to a variety of different pathogens and even non-pathogenic triggers, right? And also we know that the nature and density of pathogens varies a lot across locations and across times and whatnot, right? So it's actually very, very hard to establish direct links between particular selective event and attribute that to a particular pathogen. We have some examples, of course, for malaria, but those are relatively rare. So we wanted to start thinking about, is there a particular event in history that we could potentially try to formally ask the question as to which extent we have evidence for selection for it. And the obvious one that comes to mind is the Black Death in the 14th century, which, as you all probably know, was caused by this pathogen called Yersinia pestis, and that killed 30 to 50% of the population in Europe. So we start thinking about this. So there was this kind of obvious hypothesis that is, if the Black Death was not an important selective pressure, so the variants are neutral, the expectation is that if you were able to collect samples and characterize genetic diversity on individuals that lived pre-Black Death, that died from Black Death, and that lived right after the Black Death, the expectations that if this time window is relatively short, say 10, 15 generations, you will not expect to see much of a fluctuation in allele frequency for those variants. In contrast, if you have a variant that, for example, imagine that was protective against Black Death during the pandemic, you would expect these variants to increase in allele frequency more than expected by chance when you compare pre-Black Death and post-Black Death, because obviously the post-Black Death people are those that survived the event. So we decided to kind of try to formally test that hypothesis, and this is work led by a postdoc in my lab, Torres Vilgalis. And to do so, we basically had access to 321 ancient DNA samples from 2 European populations, like I said, before, during, and after the Black Death. So the ancient DNA work was led by Hendrik Poinar at McMaster and a PhD student in his lab, Jen Klunk. And the data set that we collected is the following. So this is a capture-based approach. So we targeted specifically a panel of immune genes, so 350 immune genes. In addition, we added to those a panel of about 400 SNPs that had been associated with immune-related phenotypes, so GWAS loci. And then we have what I call neutral loci, that are just 200 regions across the genome that are putatively neutral as they are far away from genes, et cetera, et cetera. And we're going to use those neutral loci to establish our null expectations about how different should be the levels of differentiation before and after the Black Death. So this is kind of the first result. So what I'm showing you here in the x-axis it's basically, you can think about it as like, how differentiated are the SNPs, how extremely differentiated are SNPs when you compare before and after black death. And the y-axis is the fold enrichment with respect to null expectations. And in this case, the null expectations are based on these neutral sites. So we just kind of ask whether our targeted sites, and this will be the immune loci and the GWAS loci, tend to be more differentiated than expected by chance, just based on neutral expectations. And as you can tell, in the London cohort, we do see a very strong enrichment of highly differentiated sites among immune loci. And this we then try to replicate in an independent cohort. So similar samples, similar study design, but now in Denmark. So these samples are less well dated, so it's less clean, but yet we still see again this enrichment of highly differentiated sites as compared to neutral expectations. So this is kind of already an indication that there's evidence that Yersinia pestis and the black death was an important selective pressure during the black death. And this is kind of a Manhattan plot that kind of tries to summarize those results. So across the genome, we identified 245 variants that are highly differentiated within London. And I'm highlighting in orange, these orange dots are 4 variants that we were able to replicate in this independent cohort from Denmark. So, and I'm going to show you, kind of just zoom in on those 4 variants. So I'm showing you here in these plots, it's just in blue, it's the Denmark cohort. In London is the London cohort. In red, sorry, in red is London cohort. And what I'm showing you, it's for example, for the first locus, ERAP1 and ERAP2 variant, there's the allele frequency before black death. Then in the middle is the allele frequency on people that we believe to have died from black death, and then after the black death. And you see that consistently in the both cohorts, this variant has increased in allele frequency when you compare before and after black death. And that kind of the same thing across all the different variants. So I'm going to go and enter more into the details of these variants later on. So I'm not going to spend much time on it now. But the point being that these are our top four candidates. So we wanted to understand, is there any evidence that these things are functionally relevant? So what we did is that we took cells and we started the macrophages from a panel of 30 individuals or 33 individuals that we infected with Yersinia pestis for 24 hours and then just measured gene expression levels. So this is work done by Mary and Anne in my lab. So of course, as you can tell, they look like, they are going to space because they have to work on the level 3 facility. And the first thing we asked was, are the genes nearby these locus that we identified as being our strongest candidates responding to Yersinia pestis? And as you can tell, basically almost across the board, all these different genes that are nearby these locus are differently expressed in response to Yersinia pestis, suggesting that they might play a role indeed in the immune response to pestis. But what we really wanted to know is whether these SNPs in particularly were associated with variation in gene expression of these genes, because really what we care is not just whether the gene response, is whether depending on a genotype, you have a difference in response. And what we found was 2 EQTLs, and I'm going to kind of focus specifically on ERAP2 because everything that I'm going to tell you after it's for ERAP2, and it's by far the strongest association that we see. So you see that for ERAP2, we found this very strong EQTL that is shared across both non-stimulated and Yersinia pestis infected cells, whereby individuals with the C allele, so the C allele is the protective allele, tends to have much higher levels of gene expression of ERAP2 as individuals with the T allele. So interestingly, this variant had already been previously described, and had been previously described as showing actually evidence for long-term balancing selection. So this is a variant that is that leads. It's a splicing variant that leads to a premature stop codon. So in other words, basically what's happening is that if you have the protective allele, you have a functional variant of ERAP2. If you have the detrimental allele, you basically don't have any functional ERAP2 because you have this premature stop codon. So it's a very clear and clean phenotype here. So what are these genes? So ERAP1 and ERAP2 are aminopeptidases that work synergistically to trim peptides to be so that they can be presented by MHC class 1 to CD8 T cells. So the hypothesis now is that depending on whether or not you have URAP2, you might be able to present a different repertoire of Yersinia pestis derived antigens to CD8 T cells. So unfortunately, it's not easy to come up with an experiment that will formally test that. So I mean, at this point, I'm just speculating that that might be happening. But what we do know, at least, is that CD8 T cells are really important in the immune response to Yersinia pestis. Indeed, if you infect mice that are CD8 depleted with Yersinia, you see that they die way faster than wild type mice. So this is going to suggest a potential mechanism, but we haven't formally tested it yet, at least. But URAP2 has also been shown to have non-canonical functions that, for example, are associated with variation in gene expression in cytokine responses. So what we did was to infect monocyte-derived macrophages from 25 individuals with the Yersinia pestis and measure cytokine secretion and see if those cytokine levels vary as a function of this URAP2 variant. And we, to our surprise, to be honest, I was not expecting to see anything, but to our surprise, we do see actually a very striking relationship, at least for these 4 cytokines, between genotypes for this URAP2 variant and cytokine levels, suggesting that there's an important regulatory effect of this URAP2 variant in the immune response to Yersinia. So then we wanted to push this a little bit further and ask whether this variant could also be associated with variation in the ability of these macrophages to control bacterial growth. And we did see this relationship where indeed, if you have the protective haplotype, if you are homozygote for the protected variant, you are better off at killing the bacteria. So the y-axis is the normalized percentage of bacteria that's been killed. And you see this linear relationship that the more copies you have of the protective allele, the better your macrophages seems to be at controlling bacterial growth, further supporting indeed a role of this variant in protection against Yersinia pestis. So kind of some take-home message of this story. So we see that black death seem to have indeed shaped genetic diversity around immune loci. We speculate that this comes from a different ability of APCs and chain-presenting cells to present antigens to CD8 T cells, and potentially, as I showed, differences in the ability of the macrophage directly controlling intracellular bacterial growth. And then the other thing that I would like to highlight is that, coincidentally or not, I don't know, but 3 out of the 4 variants that are our strongest candidates correspond to genetic variants that have also been associated with increased risk for inflammatory disorders or other infectious diseases. So for example, the URAP2 variant, the same variant that protects against Yersinia pestis, is a variant that is associated with increased risk for Crohn's disease. The same for CTLA4, so the protective allele, it's also associated with increased risk for lupus and rheumatoid arthritis. So I think these data more generally highlight the contribution of historical pathogens to present-day susceptibility towards modern diseases, whereby you have these variants that were selected in the past as they were apparently protective against this pathogen, that at the same time seem today to be associated with increased risk to disease. And I'm running late, so just finishing to thanking all the people that actually did the work. So I'm highlighting with these boxes those that were more directly involved in the 2 papers that I talked about more in detail. I just like to mention the strong collaboration with Guillaume Burke and Tommy Pastinen at McGill for all that was the epigenetic work, which was funded in part by this International Human Epigenome Consortium. And then I'm listing all the different collaborators, and happy to take any questions.
Luis, it's Jimmy. This is a phenomenal talk. Gosh, we have so much to talk about, but maybe just 2 quick questions. The first one is, your MOI for the flu study was it 0.5?
I think it was 0.5, but now I'm not 100% sure. I don't want to lie to you.
Yeah, so I mean, it seems low, right? I mean, are you just capturing interferon response, or are you capturing maybe you're probably not capturing a lot of antiviral responses that's independent of interferon, is what I guess I'm wondering.
I mean, so what I can tell is that we have definitely the virus is getting to the cells, specifically in monocytes. We see a lot of viral transcripts. So and we do see, like, like I said, we actually measure PFUs, like at 48 hours pausing, well, 24 and 48 hours pause infection, and we are, clearly you're getting productive virus coming out from those cells. So yeah, so I mean, we are definitely seeing a very robust response. I think that's probably the best I can tell you, is that the virus getting inside the cells, definitely. We didn't want to go with like a super large MOI, just for the obvious reasons that then you start mostly measuring cell death, right?
Yeah.
So it was a kind of trade-off. So we did a first, like, pilot where we tried to like play with dose and time points, etc., to avoid just killing the cells, basically.
The question is actually related to ERAP2, which we should probably follow up on. But the question for you here quickly is that the protective variant, is that on the NMD haplotype, or is it on the full length haplotype?
So the protective variant is in the full length. So it seems that basically having a functional ERAP2 will have been protective.
Excellent. Okay, because then we can follow up and I'll tell you about what the other variant, the other haplotype does.
Okay, yeah, for sure.
It's under balancing selection, right? Which means that,
right.
So I think there could be a really cool story, and we have some new tools that might be useful for you guys.
Okay, for sure. Yeah. I'm happy to talk about it for sure. Yeah.
Yeah, thank you so much. That was a really wonderful talk. I think I learned a lot. I don't know if we have time for maybe another one. Quick question. So I guess something that I was thinking about when you were presenting your ancestry-specific work, and particularly in the flu context, I wonder if there's a way that we could test whether all of these EQTLs that you've identified could in some ways explain differences in response to the flu vaccine on the population level. I don't know if it would make sense to look at, and I think it would be hard to find maybe the right data set to test that in, because I think that laboratory confirmed influenza is not really something that gets tested a lot, but I think that could be maybe interesting to look at.
Yeah, I mean, absolutely. I think that's probably when thinking about like how would one try to kind of translate this into a kind of a more in vivo model, because like now literally we're just hitting cells in a petri dish. That's kind of the obvious thing that we've been thinking about, and it's trying to think about it in the context of a vaccine response, which is probably the closest that you can get to. You're not going to do a human challenge with live flu. I mean, people have done it, but I'm not going to go there. So I think that makes a lot of sense. We've done that in other context work that I didn't talk about, in monkeys, etc., where we did like vaccine challenges, and I think doing that in humans will be interesting for sure. Yeah.
And also just for I guess maybe a bit of a technical question. So the cells were cultured. So for the epigenetic work, was there ways that you could control for the fact that culturing cells might have an impact on their epigenetic profiles? That's something that you could account for.
Yeah, so you're absolutely right. So the cells, specifically macrophages, so in contrast to PBMCs, where basically PBMCs is just to kind of isolate them and you work on them right away. Macrophages are a little bit more tricky, because you actually need to first isolate monocytes that are in circulation and then derive the monocytes into macrophages in culture for about 7 days. So you're right that during that differentiation process, it is possible that you actually end up erasing a lot of the epigenetic variation that existed at the beginning. And I don't have a good answer to you, because we don't really know, but that could also potentially explain why we are seeing that more of the epigenetic variation appears to be genetically controlled, because of course the variation that is genetically controlled is going to remain. And actually you could argue that actually you increase your power by getting rid of like difference in the environment that might exist a priori, right? But then in culture you kind of get rid of those, right? And therefore you are rich for genetic effects. So it is, you are absolutely right. And I don't have an answer to you.
Yeah, but as you said, I think you're still capturing like it affects like the overall variance, but you should be able to still pick up the genetic signals.
Right.
Not be
Exactly. Genetic signal should be there.
Yeah. Okay. I mean, I probably have other questions, but I think we're already past the hour. So unless anyone else has a quick question, I think maybe we'll just end it here. Okay. Well,
Thank you so much, guys.
Yeah, this was wonderful. Thanks a lot.
My pleasure. Hopefully next time I can come in person and
Yes.
just have time to chat with everyone.
Absolutely. Thank you. Bye.
Thank you, guys. Bye.